Cells are the smallest structural unit of the human body that possess all the functions and activities that biologists describe as life. Since the 1800s, there has been a great deal of research done on cells because our bodies are comprised of trillions of them. If cells fail to function properly or die, it is possible that the tissue will be damaged and may die. Because tissues make up the organs, the organ may cease to do its job, and the entire organ system's role in the body may shut down, leading to widespread homeostatic imbalance, which could ultimately lead to the death of the organism. Therefore, we will look at some basic features of a generalized animal cell. Your textbook and Mastering A and P give you much more information than we will cover here, so you can refer to these resources for more information or review activities. Although we will use a generalized cell for our descriptions of cellular components, it is important to keep in mind that our bodies are made up of 200 different kinds of cells. As seen in the diagram, each cell type has a unique structure and function in the body. As we established previously, the cell is the smallest unit of structure that meets all the qualifications of life. This slide is a review of the major organelles of a generalized or composite animal cell. Cells consist of three major compartments: 1. Plasma membrane equal outer cell membrane; 2. The cytoplasm, which consists of the cytosol fluid component and organelles; 3. The nucleus, which contains the genetic material. The organelles are the little organs of the cell. Just like our organs play a special function for the body, such as respiration performed by the lungs, each organelle plays a role in the normal functioning of the cell. There is a system of internal membranes, the endomembrane system, which includes the endoplasmic reticulum rough and smooth, the Golgi apparatus, secretory vesicles, and lysosomes, and the membrane surrounding the nucleus. The rough endoplasmic reticulum RER is involved with protein synthesis. The other form of endoplasmic reticulum lax are ribosomes, so it is called smooth endoplasmic reticulum, or SER. Although the SER is continuous with the RER, it plays no role in protein synthesis because it lacks ribosomes. Instead, the SER is involved with lipid metabolism and synthesis of cholesterol and steroid hormones. It also plays a crucial role in detoxification of drugs, alcohol, and other toxic substances. If you don't know the function of each of these organelles, refer to Chapter 3 in your textbook and / or Mastering A and P. We will begin our description of the organelles by starting with the plasma membrane and working our way through the cytoplasm to the nucleus. Our current idea of plasma membrane structure is called the fluid mosaic model. The plasma membrane consists of a double layer of lipids with embedded proteins. The lipid bilayer is made up of about 75% phospholipids. Phospholipids are characterized by have a polar head, which means it is water-loving hydrophilic and can interact with the water component of body fluids. The other part of the phospholipid molecule consists of two fatty acid tails, which are nonpolar and hydrophobic water-fearing. Notice that the tails of the fatty acids point inward toward each other, whereas the polar heads are facing the watery environment inside and outside the cell. Thus, the plasma membrane effectively separates the intracellular fluid ICF from the extracellular fluid ECF. About 5% of the membrane lipids are glycolipids, which have a carbohydrate, E group, attached to them. Another 20% of the lipids are cholesterol. Cholesterol stiffens plasma membranes, whereas phospholipids make them more fluid. The integral membrane proteins play a very strategic role in selectivity. They are inserted into the lipid bilayer, and many go all the way through it from inside to outside. These are called transmembrane proteins. Most of the integral membrane proteins play a role in transport. They can serve as pores or channels, as carriers, receptors, or as enzymes. Peripheral membrane proteins attach loosely to the integral membrane proteins on both the inside and outside of the plasma membrane. The glycoproteins on the extracellular surface help to form a glycocalyx, important for cell reinforcement of cell structure, adhesion, recognition, and other functions. The proteins on the intracellular surface are part of a cytoskeleton that helps give support to the cells among other functions. The plasma membrane provides both structural support for the cells, as well as playing a dynamic role in cellular activity and regulation. It serves as a physical barrier. This is a critical function because the physical-chemical makeup of the cytoplasm is different from that of the extracellular fluid bathing the cell. Therefore, the plasma membrane is crucial for maintaining homeostasis at the cellular level. It also regulates exchange and interaction with the external environment by controlling entry and exit of nutrients, ions, water, and wastes. Finally, it is the plasma membrane component of cells that is able to sense and respond to stimuli. The plasma membrane also has the ability to store potential energy so that the cell can do work when called up. In all living cells, the inside of the plasma membrane is negative and the outside is positive. This polarization of charge allows the cells to store potential energy so that the cell can do its job. We can liken the electrically polarized plasma membrane to a battery. Both the plasma membrane and a battery have a positive end and a negative end. They are both polarized. By keeping the positive and negative charges apart plasma membrane in cells, both the cell and battery are able to store potential energy. When the cell is stimulated or the switch is thrown on, the charge flip-flops. This provides the energy for a cell to do certain types of work, or for a battery to power a light bulb. Resting membrane potentials of different cells range from -50 to -100 millivolts MV. Changes in the transmembrane potential are behind muscle contraction, secretion of glands, and transmission of information in the nervous system. The diagram on the bottom left shows part of a cylindrical cell, perhaps the axon of a neuron. Notice the charge difference across the plasma membrane and the differences in the intracellular and extracellular ion concentrations. It is mainly the leakage of potassium and the presence of negatively charged proteins anions equal A that cause the difference in charge. The bottom right diagram shows a greatly magnified view of a piece of the membrane. Notice how potassium leakage out is much greater than sodium leakage in. Also note that the sodium-potassium pump is working to maintain the transmembrane or resting potential of the cell by exchanging 3 sodium ions out for 2 potassium ions in. Many of the activities of the plasma membrane, such as maintaining the resting membrane potential, require input of energy in the form of ATP. Mitochondria make ATP, which is a chemical form of energy that cells can use. ATP is what powers most the metabolic reactions that keep us alive and powers activities such as muscle contraction. Mitochondria have an outer membrane and an inner membrane. The inner membrane is arranged in a series of folds called cristae. The internal cavity of the mitochondrion is called the matrix. The elaborate folding of the inner mitochondrial membrane to form cristae greatly increases the surface area of the membrane into which are inserted enzymes that catalyze chemical reactions collectively known as cellular respiration. In cellular respiration, oxygen is used to make ATP from catabolism of nutrient molecules, such as glucose. The rough endoplasmic reticulum has ribosomes attached to its membranes. Ribosomes are involved with protein synthesis. The proteins synthesized by the ribosomes of the rough endoplasmic reticulum RER enter the internal space defined by the folded membrane cisternae. After some modifications, they are packaged into vesicles and make their way to the Golgi apparatus. Peroxisomes bud off the RER. They contain powerful enzymes called oxidases and catalases. Their most important function is to neutralize free radicals, which can destroy the molecular components of the cells. The RER is also the source of the integral membrane proteins of the plasma membrane, which play an indispensable role in cell transport. The Golgi complex, or apparatus, is located near the nucleus and can be quite extensive in cells with high secretory activity. The Golgi apparatus receives newly synthesized proteins from the RER. The proteins are condensed, chemically modified, sorted, and packaged for delivery to various destinations. Proteins are sorted into those that will leave the cell as a secretory product, such as digestive enzymes, hormones, or antibodies depending on the type of cell, or will stay in the cell as lysosomes, which contain powerful degradative enzymes and are involved with intracellular digestion. The illustration above shows the steps involved in transporting proteins from the RER cisternae to the Golgi apparatus, and how the Golgi complex sorts and packages the proteins for secretion or to form lysosomes or to be incorporated into the plasma membrane as integral membrane proteins. The cell has a network of internal scaffolding made up of various kinds of proteins. Collectively, the scaffolding is called the cytoskeleton. The cytoskeleton functions to help give shape and support to the cell, but it also plays a role in moving organelles or molecules from place to place within the cytoplasm. The cytoskeleton also can extend from the main surface of the cell as cilia or flagella. These structures aid in movement of the cell sperm or movement of materials along the surface of the cell cilia. Cilia and flagella have a similar makeup. Both have a 9+2 arrangement of microtubules surrounded by plasma membrane. There are 9 doublets of microtubules around the circumference, and a set of doublets in the center. The doublets have motor proteins attached to them which aid in movement. The main difference between cilia and flagella is that flagella are longer and usually single. Flagella move entire cells. The only example of a flagellated cell in humans are sperm cells. Cilia, on the other hand, are short and extremely numerous. They line surfaces of epithelia and beat in a coordinated manner to move materials from one location to another. Both cilia and flagella have a basal body at their base. The 9 outer doublets of a cilium or flagellum extend into a ring of triplets of the basal body. Ciliated cells line the respiratory tract and the uterine tubes in females. In the respiratory tract, debris that we inhale when we breathe in is trapped in a layer of mucus. The cilia beat in a coordinated manner to move the debris-laden mucus in a direction away from the lungs, where we can cough it up or swallow it. The ciliated cells lining the uterine tubes beat in such a way as to create fluid currents in the pelvic cavity. There is no direct connection between the ovary and the uterine tube, so when ovulation occurs, fluid currents set up by the cilia sweep the oocyte into the uterine tube and ultimately into the uterus. In addition to the genetic material, the nucleus contains a nucleolus, which is the site of ribosome synthesis. Ribosomes function in protein synthesis. The outer part of the nuclear envelope is continuous with the rough endoplasmic reticulum. The ribosomal subunits leave the nucleus through pores in the envelope and become either free in the cytosol or attached to the membranes of the RER.